Hi, welcome back to my channel. In this video, I will be going through like some of the important points of general microbiology. So it's like an active recall and you can do it with me as well. So first of all, in general microbiology, I've covered most of it in this video. So like this will be beneficial for you as well. So first of all, we'll talk about the contributions of Pasteur. So Pasteur contributed, first of all, he's called as the father of microbiology. Then he basically made vaccines for cholera, anthrax, rabies. Then he explained fermentation. Then he gave liquid media, which is nutrient broth. Then responsible for autoclave, then sterilization, hot air oven. Then there's one question more that they ask related to Pasteur. He disapproved the biogenesis theory, and he gave himself the germ theory. Okay. Now let's talk about Robert Koch. So in Robert Koch, you have to remember, basically he did cholera, anthrax, and tubercle bacilli. He gave solid media. He used aniline dye for contrast media, and he also showed hanging drop motility in bacilli. Then there are some Koch postulates, as we all know, like the microorganism should be in constant association, then should be able to isolate, and then inoculation should lead to the disease, and a re-inoculation or re-isolation, should like re-isolation should lead to the absence of the disease. So they are not followed by Mycobacterium leprae, Neisseria gonorrhoeae, and Treponema pallidum. So this was the question. Then these are four. Then new postulate was also given, fifth one, which was in which the antibodies were demonstrated. And then there came a modification also, which was the molecular Koch postulate, in which he said that the gene of microbe should basically satisfy all criteria rather than microorganism. So this was about the Koch postulates and various other things like molecular and fifth postulate. All right, then coming on to the next part. So Edward Jenner is basically for the first vaccine for smallpox. Then Joseph Lister is known as the father of antiseptic surgery. He gave carbolic acid. Then Hans Christian Gram for gram staining, Ernst Ruska for electron microscope, Alexander Fleming for antibiotics through penicillin, and T. pallidum was found by Shorden and Hoffman. Now they sometimes ask the resolution of human eye and so on. So human eye is 0.2 mm. Light microscope is 0.2 micrometer, and electron microscope is 0.2 to 0.5 nanometer. Then opaque disc is used in the opaque disc and diffraction plate and annular diaphragm. This is used in phase contrast microscopy, and opaque disc is used in dark microscopy. Then acridine orange was is used as in fluorescence microscopy for plasmodium, and auramine phenol is for TB. Autofluorescence is shown by cyclospora. Jumping jacks of microbiology are mycoplasma. India incornigrosin strain is used for cryptococcus. McFadden test is for Bacillus anthracis or anthrax. Then Quilling reaction is shown by H. influenza and Streptococcus pneumoniae. endotoxin gram negative, 1 to 2 layer gram negative, gram positive, teichoic acid gram positive, more lipid content gram negative, also amino acids more are present in gram negative. And here there's catch, aromatic amino acids are seen in gram positive. Okay, then more lipid content. All right, monotrichous. So mono, you have to remember PVC, which is Pseudomonas, Vibrio, and Campylobacter. Then amphi, you have to remember ALA. So amphi, ALA. So we have amphitrichus में there was a name glat allergen something. So first by L you can remember Listeria, L caligens, I think Ficalis, I don't remember, I'll have to cross check and I'll verify it and like tell later on. Then coming to LOFO, in Lophotrichus, so with LOFO we have to remember LHS. So we have Heliobacter and Spirilium. So first of all, monotrichus is something like this, amphitrichus is 2, Lophotrichus is this, then peritrichus is all around. So peri, you have to remember Perks, so E, so P for peritrichus, E, Enterobacteriaceae, except, now you have to remember the S, except Shigella, and K, so and Klebsiella. Okay, now peritrichus, as you know, all around. Then endoflagella, just one example you have to know, spirochetes. Now let's talk about motility. Now tumbling motility is shown by Listeria. Now here also at 25 degrees Celsius it is motile, but at 37 degrees Celsius it is non-motile. Then gliding motility is by Mycoplasma, stately Clostridium tetani, twitching Trichomonas vaginalis. Then falling leaf motility, Giardia, darting or shooting star, Vibrio, swarming Proteus, corkscrew is by spirochetes. Then sometimes they ask us about the inhibitors of swarming. So they are alcohol, then agar, azide, then from B, boric acid, then from C, charcoal, hydrate, then from S, surface active antigens. Then gram staining, now you know that there is primary stain, then there is a mordant, then there is a mordant, then there is a decolorizer, and then there is secondary stain. Okay, this much you know. So for primary stain we have crystal violet, then for mordant we have iodine, then for decolorizer we have the acetone or alcohol, and for the secondary stain we have safranin or carbol fustian. Okay, now coming on to bipolar staining. Now bipolar staining, they ask like which pattern is seen, so it's like favorite question, so you have to know its safety pin appearance, and it is shown by a number of organisms like Yersinia pestis, then Campylobacter, granulomatis, then H. deuceri, then then pseudomylae. All right, then Zn staining, again you should know that like there are primary stain, a mordant, then decolorizer, then so in this Zn stain, so we have first we have a primary stain as carbol fustian, then we have heating, as mordant. Then decolorizer is H2SO4, and then we have the malachite green. Now here I want you to remember a few things. See, this Zn staining is like used for a lot of bacteria, right? So what happens is there's a just the difference in the concentration of H2SO4. So if suppose we have MTB, so for that 25% is the concentration. If we have M leprae, so we have 5%. Then most of the parasitic organisms that have the characteristic of Zn staining, like hydrated cyst, Tinea saginata, then some other also, like not parasites but bacteria only, so nocardia, cyclospora and so on, they just have 1%. Then you can also do the Zn staining on bacterial spore. For that the concentration of H2SO4 is 0.2 to 0.5%. So like you just have to remember this much for Zn staining. Then Vibrio cholerae is acidic or alkaline, so its alkaline pH is in which it grows nicely. Then Albert staining. Now Albert staining, again you have to know that you have two stains. One is the Albert stain 1 and then Albert stain 2. So 1 you can remember as GAMT. So in this, first you have glacial acetic acid, then you have by A you have the alcohol, then malachite green, and by T we have toluidine blue. Then this is like Albert 2 stain has put iodine and potassium iodide. Now triple antibiotic test, here you have GCL, so you have glucose, JSL, so sucrose, and then you have lactose, and all of their ratio is 1:10:10. Now see, if we have acid it will be yellow, and if we have just give me a second, if we have yellow it means acid production is there and we denote it as A. If it is red means you don't have acid production and it is R. All right, so if the result is coming as K by K, so what do you understand by K by K is that there is no acid production, and if it is K by A you have acid production, and if it is A by A you like some amount. Then but here you have proper acid production. So in this now there's something like slant and base, so you have to know that. So suppose I'll just explain it in brief. See, you will have 3 test tubes, okay, and so what will happen is see, this is slant and this is our base, okay. So see, there can be either full on red or red and yellow, and some can be fully yellow. So if it is fully red, so it means that no acid production is occurring, so that means that it is a non-fermenter, right? So just give me a second. So if it is a non-fermenter, so you have to know the examples. So it can be Pseudomonas and Actinobacter. If it is showing some amount of, so it's showing glucose fermentation, and in this you have to remember Salmonella and Shigella. All right, then AA, acid production is there, so basically it's fermenting more than 2 sugars fermented. Now this is E. coli and Klebsiella. Okay, now let's talk about this the graph. So if you remember, graph was something like, the graph was, first we have lag, log, then stationary, then death. Okay, so this is it. Now let's just talk about the characteristics. So see, first in lag, most commonly asked question is what is seen in lag. So in this you have maximum cell size and there is an increase in the enzymes and the metabolites. All right, then in log phase you have exponential growth, and one characteristic is that in this you have uniform staining. Stationary phase, all the nutrients have been used. All right, and in this you will see that there is formation of the granules, spores, exotoxins, and so on. Involution is death. Now psychrophilic is this is basically related to all the temperature conditions. So psychrophilic is less than 20 degrees Celsius, mesophilic is 25 to 40 degrees Celsius, thermophilic is 40 to 70 degrees Celsius, and hyperthermophilic is more than 70 degrees Celsius, which is seen in Geobacillus thermophilus. And psychrophilic may you can remember Pseudomonas. Now most pathogenic bacteria are in this because it's our body temperature most pathogenic. Alright, now let's talk about transformation. So transformation idea was given by Griffith. Basically in this there is an uptake of free or naked DNA, and it is seen in Streptococcus pneumoniae. Now transduction is shown by a bacteriophage. This is related to plasmid. Then lysogenic conversion seen in Corynebacterium diphtheriae. Conjugation is mating plus you have a conjugation tube. Sometimes they ask, it was given by Liederberg and Tatum. Alright, so if they are asking about the antibiotic, so it's due to conjugation. Then plasmid versus episome. So circular DNA in cytoplasm is plasmid, and plasmid DNA plus host chromosome is episome. Then qualitative and quantitative antibiotic sensitivity testing. Qualitative has Kirby-Boer and Stokes. So qualitative is used for healthy patient, whereas quantitative is used for immunocompromised patient. And in quantitative you have dilution methods. Here these are diffusion methods. Culture media used most commonly is the Mueller-Hinton media. Then inferences. If there is no zone, so there is no sensitivity. If there is 14 mm zone, so it is intermediate. And if it is a 38 mm zone, it shows that the organism is susceptible to the antibiotic. E test, this is recent, it is called as the it's called as the epsilometer test. So it's like combination of dilution plus diffusion. In this you use an inert carrier strip. Then coming on to chocolate agar. So chocolate agar, when blood agar is heated at 70 degrees Celsius, you have the formation of chocolate agar. Now this chocolate agar is used for H. influenzae. Alright, then the Lopheral serum slope is by Corynebacterium diphtheriae. Algae medium is Mycobacterium tuberculosis. Tetrathionate broth for Salmonella. Then we have TCBS for Vibrio cholerae, and this is for Shigella and Salmonella. Then blood agar is for Streptococcus. Then acid fastness depends upon the content of mycolic acid plus the integrity of cell wall. And now we're talking about the volutin granules. Now these volutin granules are important. They basically the function is they reserve intracellular phosphate for nucleic acid synthesis. And growth condition, they are basically they grow in periods of starvation or in nutritional deficiency. So this is like most of the general microbiology covered. So yeah, so if you found this video helpful, do give it a like. And yeah, I'll just explain this just once because I think I skipped it. So see, here you have red, all is red. So this is K by K. Now here is acid production, so it is K by A yellow, right? Now here is A by A. Now this means कि कोई acid बना ही नहीं, so no sugar was fermented. So what is responsible is Pseudomonas actinobacter. Alright, so I'll just use it. So like Pseudomonas and actinobacter. Then what happened was there was some acid production but not totally acid production. So Salmonella and Shigella. Then total, so they're able to ferment more than two sugars. So that is by E. coli and Klebsiella. So that is why this acid is formed. All right. So yeah, so this explains most of it, and I covered most of the general microbiology. Like mostly, I have covered all the microbiology that I think is like that was in my notes. If you like the video, do give it a like, share, subscribe.